Это генетик из университета штата Джорджия, факультет клеточной биологии, член научного комитета Международного фонда Аниридии, который уже больше 19 лет занимается изучением гена PAX6.
Good morning. Thank you very much for the invitation, and I apologize that I was late. It's an entertaining story, but we'll leave it for later. By now you've seen most of this information. Aniridia, the one point on aniridia that I'd like to bring to your attention is that, okay, so what I'd like to bring to your attention is the frequency of aniridia is estimated between 1 in 40,000 people to 1 in 100,000 people. I think most of us now think that it's closer 1 in 40, 1 in 50, 1 in 60, somewhere in there. So the important thing is that aniridia affects all structures of the eye. And this is because the early in development, the eye, as you've already seen, has the cornea, iris, lens, and retina. And of course, the retina is composed of different cell types. In order to understand why these cells are all affected, we need to go back and understand a little bit about how the PAX6 gene is expressed in development. What you're looking at are some sections of chicken embryos, which we're going to use to help us understand human eye development. And so the important thing is that this is the early eye. So what you're looking at is what would be part of the brain that will take the eye. So this is the future retina, and the surface is the future cornea and the future lens. As the eye begins to develop, and this is true for chickens, mice, birds, you and me, and all creatures with all vertebrates with eyes, we see the developing optic cup, and this is the future lens. And then this is starting to be more recognizable. This is the future cornea, two distinct cell types. One cell type is the cell type that we were just looking at. So that's the part that will give rise to the corneal epithelium, which Dr. Evans was talking about in corneal dystrophies. Underneath, the red cells form the stroma. And so the stroma does not express PAX6, but it listens to language coming from the corneal epithelium, which does express PAX6. And so the important thing is that the cells that use PAX6 to do their function are talking to all parts of the eye to make sure that it forms properly. And so I apologize that this is somewhat simple, but I wanted to make sure that we all understand what a gene is so that we can understand the next part. All genes are made up of a simple alphabet. They're chemical structures, but we don't need to worry about that. But the alphabet is important. And so we use this to visualize what our genes look like. The alphabet is A, G, C, and T. The combination of that code is very important. And so if we think about it, DNA is simply like a library. It contains all of the information that makes who we are, who we are. Every single cell in our bodies has the same genomic DNA. Every single cell in our body has 2 copies of the PAX6 gene. In the cells that use PAX6, so the cells that are affected in aniridia, the information in the library is then transcribed or written out in an architectural blueprint. And that is what we call message, and this will become important in just a few minutes. So information that's stored in the DNA is copied into a message or a form that the cells can use. What we say is that the cells express the gene, and this is a mouse embryo that you're looking at that was stained, that was made to show where the PAX6 gene is expressed. So this would be true for humans as well. So we see PAX6 expression, these dark blue cells in the eye. in the brain that gives rise to the part of the brain that we think with, the spinal cord down our backs, and the endocrine pancreas. It is the protein that is important. So DNA simply stores information. By itself, it does nothing. It's important, but it doesn't do anything in the cell. Information is then copied into RNA, and the RNA is used to assemble the protein, and it's the proteins that we actually do the work of the cell. So it's the protein that we have to pay attention to. And so very simply, genes are made of DNA. Not all of the DNA is used for genes. The same genes are in all cells, which is very important, but only the cells that need the gene are going to make the message, which is then made into protein. Genetics 101. This is simply a representation that we use to understand PAX6. The letters represent the message, so the language is still AGC, but we've changed T to a different letter, U, in the cell. For simplicity, I've left T here so that we can see it. The alphabet that makes up protein is always in groups of 3. These encode for the amino acids that make up the protein, and so, for example, ATG makes the amino acid methionine, which is an important start. That's the first part of PAX6. The reason this is important is because most of the mutations that affect PAX6 that give rise to aniridia are going to affect this assembly. So this is what PAX6 looks like to a geneticist in its alphabet code. So what you're seeing now is a picture of what the PAX6 gene looks like to us. The long blue line represents the genomic DNA, or the part of the DNA where PAX6 is located. The boxes with numbers under them indicate the exons, or the chapters that are going to go into making PAX6. So you can think of this as a book. The chapters of the book are represented by 0 to 13. There are a couple special features that I want to point out. The green boxes are the parts of the gene, the control mechanisms that turn the gene on or off, like a light switch in this room. If these are off, the gene does not turn on. So if the light switch is off in the room, the lights will turn off. PAX6, as you know, is in a neighborhood on a chromosome that has other genes, including the Wilms tumor gene, WT1, a gene called FSHB, and another gene that sits way, way over here that's called brain-derived neurotrophic factor. For people who have Wagner syndrome, this entire part of the chromosome from WT1 all the way through PAX6 and very often all the way over to here has been taken away. The next slide is somewhat complicated, but it's also important to understand. So we think about PAX6 making one protein, but actually it makes 3 different types of or forms of the protein. The top line, as we've seen before, represents the DNA in our cells. This line, and this line, and this line represent the message or the architectural blueprint. And then these two lines, these last two, represent the protein that's made. So I've shown schematically the type of protein that we normally think about with PAX6. It has three domains that are very important. We call them PD, HD, and PST. This is the form of the protein that's made by most cells. Mutations, however, can affect this form of the protein, a form of the protein that has another piece here, as well as this protein. So we're going to change how we look at PAX6 now to make a picture of the protein. The top line shows the domains PD, PB, HB, and PST as they would occur on the chromosome. This is a picture of how I think of PAX6. And so there are three parts that we have to pay attention to. The red part and the yellow part both allow this protein to bind to DNA. The green part of the protein interacts with other proteins to help PAX6 do its job in the cell. It's important to know that aniridia is caused by a mutation in only one of the two copies of PAX6. Mutations in both copies of PAX6 are lethal, and the child will die before birth. So everybody who has aniridia has a good copy of PAX6 and a PAX6 that has a mutation. And so the way that this works is that a change in the DNA represented by the red symbol is copied into the message, and it's this change which prevents the protein from working. So this is the idea that Dr. Evans was alluding to earlier. We'll skip this. Okay. So there are different types of mutations. SNP means that there's a change at one of the letters. So for example, we might change an A to a T. Shown in green are the types of changes that we call insertions or deletions. And so the new sequence has either been put into the gene or sequence has been removed from the gene. The last two represent changes in how the architectural blueprint is made, and so we call these splicing mutations or run-on mutations. This is what data looks like for our lab and many labs. And so here's the code from a person, so G, C, T, C, C. We can read out these peaks until we get to here. And so you see that there is a green, which represents A, and also a red, which represents T. That represents a change at that position that causes a mutation. And in this particular case, it occurs early in the gene. This is the most common type of change, and I'll show you that in a moment. What a nonsense mutation does, so in blue, up here, with the E there, so that's what normal PAX6 looks like. If there's a change in this letter, G to T, it creates a new word that says stop. This is a nonsense mutation. This will occur in 2/3 of all people with aniridia. An insertion or a deletion looks like this. So at this position, we now see two peaks that are going all along. And so that means that new sequence has been either added or deleted. In this case, a single T was removed. Okay. An insertion or a deletion changes the way the alphabet code is read. And so again, we have our blue with E. The G has been removed, so this is a deletion. So now our new three-letter alphabet goes G, T, A. And now it goes A, G, C, and creates a new stop. And so that makes a non-functional protein. A splice junction mutation, so now I have to explain something a little bit different. In black, these are the letters that make up PAC6. The letters over here form the, so this would be the chapter in our book. This would be between chapters. If we change that position, we can no longer put the book together properly, and the cell cannot read the volume. Once again, it changes the way PAC6 is made and creates a new stop codon. So, now that we understand a little bit about what mutations are, now that we have a language that we can communicate with, essentially, roughly 3 out of 4 people with aniridia are going to have a mutation that affects the way the PAC6 protein is put together. The remaining people are going to have either very large deletions that give rise to WAGR, or they're going to have small deletions, so taking out parts of the gene that control how the gene is turned on or turned off. In other words, it's as if we took the light switch out of the room. In our group, there's one individual that we could not find a mutation for, and there's one individual that had a mutation in a different gene. So again, most people who have aniridia, 9 out of 10 people will have a mutation in the PAC6 gene or a mutation that changes the way the gene is turned on. If we begin, so now we're going to look again. This represents the protein in the chromosome. If we ask, where do mutations occur, we see this is the start of the protein. This is the end of the protein. These bars represent the numbers of mutations that we see at any position. And you can see that there are mutations that can occur anywhere along PAC6. If we look by domain, we see that approximately 41.3% of mutations are going to occur to stop the protein from being made at this point. 15% will block the protein from being made at this point. 18% will block this part of the protein from being made. If we look at the distribution of mutations by type, so in our group, 17 had nonsense changes, so that's that single letter change that creates a new stop to prevent the protein from being made. 21 will have an insertion or a deletion, and 10 had a problem with putting the chapters of the book together. These types of mutations, the insertions and the deletions, tend to be individual, so they occur in one person but not another person. Mutations of the nonsense type can occur in many people. These mutations, insertions and deletions are probably cannot be treated by any drug. These are the mutations that are treatable by a drug. The remaining 1/3 of people with aniridia are going to have a change of something like what you see here. So again, we're representing the gene in the genome in our cells. This means that there is a deletion. So this piece of DNA is gone, or this piece of DNA is gone. And so these are very rare, or much rarer, but they all result in aniridia. These two arrows represent changes in what we call an intron. So I've been saying that this is the between chapter, so this is the region between chapters. We do not yet understand how these work. Lastly, and Tatiana will be talking more about this in her presentation, there are very large deletions that can give rise to aniridia. I want to highlight one region. So this is a very different schematic or picture than what we've been seeing. PAX6 is right here at this end. If this were 56 kilometers from here to here, this would be 160 kilometers away. The gene ends here, but changes all the way over here, 160 kilometers away from the gene, affect PAX6. And so you can have deletions that leave PAX6 alone, but still keep it from being turned on. Those are very rare, but that's what my lab studies. There are other changes in PAX6 that can give rise to diseases that are not called aniridia. This is one example. So this boy is not considered an aniridic, an individual with aniridia. He has a microphthalmia. He also has autism. When we looked at his DNA, so this is again a representation of the PAX6 gene on a chromosome. This red box is the same red box that I showed you earlier, and it was duplicated in this child. And so a duplication or an alteration of PAX6, and we know the protein is made, but the function of the protein has been changed so that it no longer does the appropriate job. And so to wrap up, normal eye development requires 2 copies of PAX6. Aniridia results in mutations that take 1 copy of PAX6. All of the cells in the body have 2 copies of the gene, every single cell. But the only cells that use PAX6 are the cells that are affected in aniridia. And these include the cornea, the iris, the ciliary body, the lens, and the retina. Additionally, there are other parts of the body that include the nose, parts of the brain, especially the ear, and the pancreas. And we know that these are also changed in individuals with aniridia. And so I would like to thank you for your kind attention. I hope that this has been helpful. The people that have been largely involved in the work include Anastasia Babalev and Maggie McDougall from my lab, And this is a collaborative effort that includes the Aniridia Foundation International, Dr. Netland and Dr. Rob Granger at the University of Virginia, Dr. Geisert and Dr. Freeman, who is in the audience back there from the Hamilton Eye Institute, and funding from the Sharon Stewart Aniridia Trust. Thank you very much.